Okay, well, it seems that it's about time that we got started with today's webinar. I'd like to first thank all of you for joining us today and attending this webinar on live cell imaging. My name is Augie Golub. I'm an R&D scientist with Thermo Fisher Scientific. I'm located in the Eugene, Oregon facility, which was formerly the Molecular Probes campus, where we work on developing many of the commonly used dyes and fluorescent conjugates that are used in fluorescence microscopy. And today's webinar will be solely focused on live cell imaging using fluorescence. And a specific breakdown of the topic will be presented in a manner where we'll cover what we refer to as the 5 steps to live cell imaging. So the 5 steps to live cell imaging will essentially serve as kind of an outline for the remainder of this talk, and the steps are outlined here in this slide, where essentially the first 2 steps are largely aimed at essentially determining whether live cell imaging is really necessary for the type of experimental question that one is trying to investigate, and then of course culturing those cells in preparation for a live cell imaging experiment. And so with those 2 steps being something that's pretty commonly performed in most labs, we'll go ahead and focus most of the content in today's webinar on the last 3 steps, which is really where our expertise from the product development comes in, with determining the optimal types of labels for live cell experiments, as well as how to optimize your labeling and imaging conditions so that you can get quality data. And then last but not least, we will also touch base on how to go about actually performing live cell imaging without any detrimental effects, because as many of you are probably familiar with, when it comes to live cell imaging, it can be quite a bit more challenging than imaging fixed dead cells. So what are some of the main advantages of live cell imaging over fixed cell imaging? So if we start at the very first step of kind of the planning phase, where we're trying to build a foundation for our entire experiment, we should really sit down at first and determine what advantages we stand to gain from moving into live cell imaging versus kind of the traditional fixed type of analysis. So of course, one of the main advantages can be illustrated with these panels here on the right, where if we look at the traditional fixed cell specimen on the bottom right, we might be able to investigate some type of biological event happening over time by taking snapshots at different time points. Whereas if we perform live cell imaging, we can actually observe these processing as they happen in real time. And so this allows us to track interactions between cells and within cells and their compartments. And so this, of course, grants us a whole new dimension for analysis and much better physiological relevance. Now, some of the considerations to keep in mind, of course, is that the labeling approaches for live cells can be quite drastically different than the approaches that one would take for fixed cell preparations. And then from an imaging standpoint, as I mentioned even in the previous slide, it can be quite a bit more challenging to actually capture this data because the cells are moving and you're subjecting them to light over continuous time periods, and they need to be in a proper environment and incubator. So there's quite a few things to think about here, and we'll talk about these in subsequent slides. So if we go back to these 5 steps, we can move into step number 2, where we really are now committed to a live cell imaging experiment. And so we need to start off with a nice healthy cell culture, because as with really any biological experiment, a successful experiment starts with a nice healthy cell culture. So we need to learn how to properly grow and maintain our cells in their optimal growth conditions. I won't spend too much time on this topic because, again, I think that this is something that's very commonly found in most labs. We have pretty standard practices for culturing and growing the cell types that we're commonly working with. Some of the more common considerations to think about when culturing your cells are, of course, the plates and the dishes that are being used, any particular additives or media supplements that might be required for your specific cell type. And then, of course, you need to have the proper environmental control for these cells to grow in an incubator. But before you can even really start to perform any types of experiments and assays, and even for routine passaging of cells, it is quite essential during the culturing phase to have a proper cell count of your cultures as you're progressing from one passage to the next, and as you're even sub-passaging from just maintaining your cells for an experiment. And again, for a successful experiment, it really does need to begin with an accurate cell count. And so cell counting is relatively straightforward to perform, or at least it seems that way on the face of it, until you start to really kind of get into these higher density cultures and being able to track viability along with total cell count. Where instruments and benchtop cell counters such as the Countess 2 FL are really very powerful companion devices that really enable you to not only get more accurate results but more reproducible results from one time to the next. So this is a type of instrument that of course allows you to not only generate a total cell count but also viability cell count in order to assess how many of the cells that you have in your population are actually viable and usable for downstream applications. Going back to our outline of steps, we will now move into really kind of the meat of the rest of this talk, which, as I mentioned in the introduction, will really be focused on the steps 3, 4, and 5, which are, the labeling, optimizing, and imaging in live cell cultures. So if we start with kind of a high-level overview of the different labeling methods that are available to us, generally speaking, they fall into these 3 pretty distinct categories, where we have perhaps the most commonly used approach here on the left, which is using antibodies. Typically, this is, of course, done with a primary antibody that recognizes your antigen of interest inside or on the outside of the cell, and then that primary antibody is followed up with a secondary antibody that's conjugated to fluorophores most traditionally, and then those fluorophores are what we are able to detect. Unfortunately, this approach is generally not compatible with live cell imaging, since antibodies tend to be rather large and bulky and not cell permeable. In the rare instance that you have a cell surface marker, you may be able to visualize it using an antibody, but most intracellular labels are not compatible with antibody labeling. So this is where we now start to rely on different types of approaches. One of them, of course, is a genetically encoded approach. So these are the traditional fluorescent protein types of labels, so GFP, RFP, and really all the rest of the fluorescent protein family. And these are, of course, require live cells in order to be expressed and folded up properly and become fluorescent. So this is very much a live cell compatible workflow. And then really the bread and butter of the campus that I work at, the former Molecular Probes campus, are of course small molecule fluorescent conjugates, where we rely on organic dyes in order to either deliver the dyes themselves directly to a compartment or conjugate the dyes to another molecule in order to give it specificity. And not all of these are live cell compatible, but many are. So we'll talk about some different examples of live cell compatible fluorescent molecules in the next set of slides. But first we'll cover various approaches towards genetically encoded labeling methods. And one of these methods that we have developed over the years is called BACMAM. And BACMAM essentially stands for insect baculovirus with mammalian promoter. And as that name implies, what that tells us is that this is a technology that takes advantage of an insect virus, a baculovirus. And what we've done is we've substituted the insect promoter with the mammalian CMV promoter in order to facilitate the expression in mammalian cells. So On the right is a table that highlights some of the commonly available BACMAM reagents that are ready to be used. And that this is really one of the key advantages to this approach, is that because this is a prepackaged virus, it is not only easy to use, but it is ready to be expressed in cells without really any molecular cloning needed by the end user. And as you can see on the table on the right, there's a pretty broad selection of both GFP and RFP fused targeting domains that either enable you to visualize different organelles and compartments within a cell, as well as different cytoskeletal structures. So one of the key considerations for using BACMAM technology is essentially getting the most optimal transduction efficiency. So the reagent is relatively easy to use, where essentially it comes prepackaged as a concentrated viral titer that you essentially dilute onto your cells and incubate overnight. And so then when you show up the following morning, you have cells that are expressing your fluorescent proteins and are now ready for live cell imaging. Generally speaking, the highest transduction efficiency happens while the cells are still in their log phase growth, and so you want to transduce before they're fully confluent, generally speaking at a confluency of about 70% or less. And then different cell types respond to the virus in different levels, and they may require a different multiplicity of infection from one cell type to the next. So this is one of the key parameters that really needs to be determined for your specific cell type, regardless of the virus that's being used. You need to determine what's called the MOI, or the multiplicity of infection, and that's done relatively easily. You essentially need to establish a titration of the viral particle on your cell, and generally speaking, this range is somewhere between about 5 and 100 viral particles per cell. And so using a very simple equation that's highlighted there on the bottom, you can determine the volume of reagent that you need for your labeling. Now, besides genetically encoded approaches, as I mentioned, another commonly taken approach is using small molecule organic dyes. And so this is what I would like to actually spend the vast majority of the rest of this talk focused on, is different types of fluorescent labels and molecules, and different approaches that we can take towards optimizing their use. So here we have an example of a live cell, a U2OS cell specifically, that's labeled with 3 different tracker dyes. So many of our trackers are indeed intended for live cell imaging. And so here what we have is an example of a cell that's labeled with 3 different live cell compatible trackers. We have a tubulin tracker, deep red, a mito tracker green, and a lyso tracker blue. So what we now see is that we're, of course, able to not only label these specific structures, but we're able to multiplex using multiple dyes in order to visualize different structures at once. And one of the really key advantages to this approach over genetically encoded approaches is the speed with which you are able to achieve labeling. This is generally accomplished within anywhere from 15 minutes to an hour. Depending on the reagent and its mechanism of loading. And another really great advantage that can often be overlooked is that because the small molecules are incubated on top of the cells in the media, what you end up with is a very uniform staining across your entire population. That can be one of the significant drawbacks to genetic approaches, is that you'll have cells that have taken up multiple or variable levels of virus and are therefore variably expressing compared to one another. So you can have quite drastic levels of labeling and expression, whereas with these small molecule fluorescent labels, the labeling tends to be very uniform because all cells are incubated with the same concentration and for the same amount of time. Some of the considerations to think about, just like with everything else, there's of course drawbacks to this approach as well. And one of the key drawbacks is that you can get non-specificity that can result from excessive labeling, is essentially from using too high of a concentration. High concentrations will also potentially affect the physiology of your cells and potentially induce prolonged cell cycle and maybe even cytotoxicity at very high concentrations. But when you get those concentrations and conditions right, they of course enable you to perform live cell imaging that can be tracked over time, either on the scale of hours or even days. So I'd like to start with really just a very high-level consideration of using small molecule fluorescent labels, and that's concentration. This is something that we spend a lot of time optimizing during the development of these types of conjugates, and so this is an example of that tubulin tracker deep red that I showed in the previous slide. And this is something that I worked on personally over the past couple of years. So during the development of this particular label, what we found is that if we tried to perform these long time-lapse experiments with cells incubated with about 1 micromolar of the reagent, what we see is that after about a couple days and at the end of 48 hours in this case, we see that the cell cycle essentially completely slows down, and at the end of this time lapse these cells die completely. Whereas if we cut that concentration down tenfold to about 0.1 micromolar, we can now see that at this 48-hour time point, the cells are not only completely alive, but they're able to go all the way out to 72 hours. And during this entire time lapse, they're able to divide, proliferate. The microtubules are still exhibiting their normal dynamic behavior, and everything is physiologically unperturbed in this event. And the only difference essentially between these two experimental conditions is the concentration that's used. So it's a very important consideration when using these types of labels for long-term time-lapse imaging, is to make sure that you do not excessively label, because the dye itself can of course interfere and perturb with the natural physiological mechanisms of your cell type. So that's really just one of the key parameters that needs to be optimized. And again, oftentimes this is something that we will spend a lot of time performing the optimization on the front end, but of course we're not really able to do this with every single cell type out there. So it is something that sometimes needs to be performed on a cell type specific basis, but we'll at least provide general guidelines on concentrations and fold dilutions on where to start. So we'll move into step number 4 then and really talk about some of the other parameters that can be thought about for optimization during live cell imaging. One of the really key parameters to think about when investigating live cells is of course this question and idea of, are we perturbing the system, and are we causing some type of toxicity to our cell line just by the sheer nature of labeling them with a fluorescent molecule. So because these experiments can be quite time consuming to generate and to really fine-tune, it's quite important to really investigate the state of the cell health upon either your labeling or some type of treatment that you're doing. And this can be done using various indicators that we refer to as cell health indicators. And so these are typically reagents that span the cell health continuum, as we refer to it as, where you have completely healthy cells, and the reporter will enable you to get an assessment of how healthy or if those cells are indeed healthy, all the way out to a completely dead population and everything in between. So what we have here on the right is an example of a couple different reagents that can actually be multiplexed to give you a combined picture of cell health, and in particular mitochondrial health, by looking at the mitochondrial health using a small molecule label called TMRM, which loads based on mitochondrial membrane potential. And this is being multiplexed with a reactive oxygen species detector called CellRox green. So what we have here is an example of U2S cells that have been treated with a drug called menadione, which displaces cytochrome C from the electron transport chain. And so what this now results in is a drastic increase in reactive oxygen species. And so if we start this time lapse we'll see that these red healthy mitochondria start to depolarize, and therefore the TMRM signal leaks out. And we now see that as the cytochrome C is being displaced, and we get this uptick in reactive oxygens from the metadione taking effect, we see that the positive signal from the cell rocks, the reactive oxygen species sensor, starts to come on. So this is a really elegant and effective way to investigate the health of your population by using a combined approach, one that gives you a readout for healthy cells and one that gives you a readout of toxic cells. Now, these types of experiments, as I mentioned, can be rather time-consuming to not only set up but also acquire, and can involve quite a bit of setup and optimization and troubleshooting. So a really easy and elegant approach towards speeding some of this work up on the front end is to use a slightly simpler approach where we're not necessarily doing time-lapse imaging and setting up these continuous multi-hour overnight potentially time lapses and trying over and over again. Is we can actually use the countess once again, but instead of just simply counting the cells like we did at the front end for culturing and passaging, the countess FL, or the countess 2FL specifically, is actually compatible with 2 fluorescent light cubes from our EVOS microscopes. And so what this now allows you to do is investigate fluorescence or a positive fluorescent signal using a benchtop cell counter. So if we go back to that same example from the previous slide of investigating mitochondrial health using TMRM and cell rocks green, if we pull samples from these various time points and simply trypsinize the cells so that we can load them into the countess, we can now assess whether the treatment is working and if the reagents are indeed giving us enough signal in the proper response by assessing the fluorescence in the countess 2FL. And so on the bottom, we see a representative image of one of those data points where we see that we're at this transition between the Texas red TMRM signal going down and the GFP positive cell rocks green signal coming on. So it's really just another tool that one should consider when really kind of starting to set up a new set of experiments. This can be rather powerful and quite valuable in terms of time savings. So in this next set of slides, what I'd like to talk about a bit are different approaches that one can take to improve the loading and retention of these fluorescent labels that we've been talking about. Because oftentimes it can be a bit of a challenge to get some of these labels into living cells, and then once they're in there, they tend to want to disperse and essentially leak out over time. And so there are a couple different reagents and approaches that we can utilize in order to improve both of these steps. So the advantages here are, of course, that it enables us to get higher signal with lower background over time. But of course, some considerations that we have to take are that these approaches may not be compatible in all cell types, and the fact that we're going to be adding additional components to these experiments also mean that potentially we may induce some cytotoxic effects. So we'll talk about that here a little bit in the following slides. The first approach I'd like to talk about is a method for improving the loading of different fluorescent labels. And so this can be done with different compounds that we typically refer to as surfactants. And one of them is called pluronic F127. In the previous slide, you saw an illustration referring to a product called PowerLoad. And PowerLoad is essentially a combination of these different pluronics that has been formulated for loading of AM esters. Here what we're using is a single pluronic, F127, that we have found to be very effective for loading our green tubulin tracker. So we'll be using this tubulin tracker example throughout much of this talk. And as I mentioned before, the new reagent that we worked on recently is this deep red version of tubulin tracker. So what we can actually see here is that if we look at our original tubulin tracker, the green on the bottom left, we have a fluorescent image that is matched in exposure and contrast to the image on the right. And what we see is that if we just incubate with the tubulin tracker by itself, we don't get very effective loading at all. The signal is actually quite dim. and is really barely above background, and that can be reflected in the quantification there on the right. But if we co-incubate and load with Pluronic F127, we see a drastic increase in the loading of this particular label. Now, on the other hand, if we employ the same approach with our new tubulin tracker Deep Red, we'll see that the molecule by itself is actually quite effective at loading without Pluronic, and that by the addition of Pluronic, we actually are causing a detrimental effect on the amount of signal that we have to work with. So this is what I was referring to in the previous slide, where these approaches aren't necessarily a universal solution and may indeed be detrimental, but in the cases where we've found them to be effective, they greatly and drastically improve the loading of these fluorescent dyes. Now, of course, another issue that you often encounter when doing live cell imaging, especially over long periods of time, is that even though the label might get loaded and is successfully internalized into the cells, you often are required to perform what's called a washout step. So that means you have to exchange the media that contains all the unbound and non-loaded fluorophores so that you can decrease the background that you're working with. And so what this of course means is that as soon as you exchange the media, you now are going to have exchange of the molecule from inside the cell wanting to leak out and go outside. So that's what we can essentially see here, is that the retention of these labels will start to suffer after a couple of hours. And so this is again another example of tubulin tracker, and this one is just looking at tubulin tracker Deep Red. And if we use a efflux pump inhibitor called probenecid, what we see is that we can actually greatly improve the retention of these small molecules over longer periods of time by enabling them to stay retained inside the cells by blocking those efflux pumps. So on the top, we have a representative image from the first time point of these cells being loaded, and then they're being incubated and imaged over time in complete media, but without any probenecid present. And what we see after about 2 hours is that the signal not only decreased, but the background greatly increased, indicating that the probe is leaking out of the cells and into the media and contributing to this background fluorescence. Now, if we perform the same type of labeling, but incubate these cells with probenecid present in complete media, what we see is that we can image over this time course of about 2 hours with really very minimal loss in signal and very minimal increase in background. And you can see this being present on the right-hand quantification as well. Consideration with this, as with many things that we've talked about so far, including the labels themselves, you want to make sure that you're not using them at too high of a concentration. The same holds true for probenecid. You want to make sure that you're not using it at too high of a concentration or for too long, otherwise you will induce cytotoxicity from that blockage of the efflux pumps over extended incubation times. Last but not least, once we've loaded the cells and have incubated them with something like probenecid to prevent the leaking out of the label, we're now of course ready to start imaging our cells on a live cell microscope. And so at this point we're going to start subjecting the cells to a fair amount of light in order to get some signal out of these fluorophores. And as many of you are, I'm sure, familiar, light is not only damaging to the cells themselves, but is also damaging to the fluorophores that we're imaging. So with many fixed cell protocols and imaging approaches, we will often use a mounting medium which prevents photobleaching, so we'll typically refer to these as antifades. And what we have developed in the past is an antifade that's compatible with live cells as well, with the idea being that we're also trying to preserve not only the signal from fixed cell preparations, but also all the signal that we get in live cell preparations. So here we have a couple representative images of cells that are being imaged at 120 independent consecutive exposures with and without the prolonged live antifade live cell imaging reagent. And on the top we have the cells that are being protected with prolonged live, and on the bottom we have cells that are untreated. You can see the drastic difference in the remaining signal with and without the prolonged live antifade. Another consideration to think about with this approach, though, is just because you're able to protect the signal from photo bleaching, that doesn't mean that you should continue to put light into the system, because even though you have signal remaining from your fluorophores, high illumination may still induce phototoxicity. We'll move on now into a different category. Instead of looking at how to label and retain and preserve signal, what I'd like to spend a little bit of time talking about is the other side of imaging, which is background, right? So everything that we do in imaging is really aimed at enhancing our signal while lowering the background in order to have the best contrast, not only for visually appealing images, but for something that we can actually quantify and segment and get some type of valuable information out of. So we have various sources of background that we need to deal with. We of course have background from the instrument and the camera or whatever detector that we're using, which is something that we rarely have any control over, but we do have control over some of these other sources, such as from the reagent itself. As I briefly alluded to a couple slides ago, when we typically do these small molecule labeling experiments, we will often need to remove and wash out the label at the end of that incubation to remove any non-specifically bound probe. And so what this now of course leads to is the leaking out of that probe out of the cells, but it does remove much of that extracellular background that will be found in the medium. The samples that we work with, of course, can also be autofluorescent, specifically in the shorter wavelengths, and the greens and the blues. And then the media that many of us use for culturing cells will have autofluorescent components that are very useful indicators of pH, but are quite detrimental in terms of image quality. So this is probably one of the simplest steps that we can take towards reducing background, is actually in the culturing media. By simply removing those autofluorescent components from the media, we can greatly improve the contrast by reducing the background from the media contribution. And this can be accomplished by using a specific formulation of DMEM that's called Fluorobright DMEM, which is essentially DMEM media but without the phenol red in it for indicating pH. So what we can see is a couple representative images of regular DMEM with all the components, including the phenol red, and then on the bottom we have a representative image of cells that are being imaged in this optically clear Fluorobright DMEM. And then on the right is a quantification of this of signal to background in either PBS, which of course is positive control here, that is also optically clear, that is essentially comparable to the Fluorobright DMEM signal to background, whereas DMEM with phenol red has relatively low signal to background compared to these two approaches. So this is a pretty simple thing to do, and it can really greatly improve the signal to background by eliminating background from the media contribution. Now, another approach that one can take specifically in live cells is by use of a background suppressor. So this is achieved with a different reagent that's called backdrop background suppressor. And the way that this works essentially is that it's a reagent that quenches any signal outside of living cells. And so it's a dye that's a general quencher of fluorescence, but it is not cell permeable. So what that means is that when you incubate cells with a label such as tubulin tracker green or tubulin tracker deep red, again going back to that same example of these two molecules that we've been talking about for loading and retention, what we see is that if we don't perform a washout step and we take an image with tubulin tracker green in particular, we see that every single pixel in this frame is fully saturated. And then the same is essentially true for tubulin tracker deep red, but to a slightly lesser extent. Now, if we start to take a live movie of this acquisition and we flow in the backdrop suppressor, what we see is that as soon as the backdrop is applied and incubated with these cells, it successfully quenches all signal outside of these cells. And so again, the idea here is that it's a general quencher of fluorescence, which is not able to cross the cell membrane. And so it quenches all the unbound dye that's in the media while leaving your intracellular signal intact. And so this works very well with many green and red fluorescent conjugates, and then as you can see here, to a certain extent, it's also quite effective with deep red fluorophores. But again, this is specific to living cells only. In the event that the cell membrane is in any way compromised, whether that be due to cell death or due to some type of fix or permeability type of workflow, the quencher will get inside the cells and it will quench all signal equally, inside and out. So it is only for live cell imaging. So that was a pretty a brief overview of some of the different labeling methods, as well as some of the different optimization methods. And so then in the last bit of this talk, I'd like to spend a little bit of time talking about different approaches and consideration when it comes to actually imaging live cells on a microscope. So first and foremost, much like with step number 2, where we needed to culture the cells in preparation for a live cell experiment, when we're performing a live cell experiment, especially over longer periods of time, one of the key considerations to keep in mind is that we need to keep the cells in their natural, healthy environment. So this means that we need to incubate them. Generally speaking, this is done at 37 degrees Celsius with about 5% CO2, 20% O2, and then we try to keep the humidity as high as possible. And the idea behind keeping the humidity high is that you prevent evaporation of your media over time as it sits on the microscope. The higher you can keep the humidity, the less evaporation you'll have, and the longer you can image. So here we have an example of 48-hour time lapse, taking images every 2 frames on one of our EVOS M7000 microscopes. And so this EVOS M7000 is equipped with an on-stage incubator that you can see there to the left of the microscope. And so that essentially provides a small incubator on the stage of the microscope that enables you to not only modulate the temperature that the cells are in, but also the environmental condition in terms of gas content and humidity. But if we keep things generally normal and constant, what we can now see is that we can monitor cells over time, and we can see these cells move and divide and proliferate over the course of this time lapse. Now, in order to generate that type of data, it is important to, of course, keep your image in focus, and that can actually be pretty challenging when it comes to imaging live cells, because live cells are still alive and therefore they're moving. And they're not only moving moving laterally or in X and Y, but there's also slight shifts in their Z position based on small adjustments and small shifts in the temperature, as well as just the potentially even the stage of your microscope, depending on the type of instrument that you're using. And so this is where we of course need to use some type of auto-focusing routine in order to keep your image in focus, and most commonly this is performed by using the fluorescent signal itself. As we'll run an algorithm that will take a small z-stack through a volume, and then that z-stack will interpret where the signal is essentially the highest in order to keep that signal in focus. But what this results in is a whole lot of extra light going into the system and exposing to your cells. And so this can actually result in a fair amount of phototoxicity. And this isn't really something that's only a problem for autofocusing. It's a general consideration for live cell imaging, to keep in mind how much light and how frequently you're sampling, because if your sampling frequency is too high or using too much light, you'll essentially end up with a lot of cell death. And so what we can see is an example of that here on the left is if we perform autofocus with high illumination settings, we'll see that at about the 20-hour time point, most of these cells stop moving and indeed start to completely die by the end, whereas the same time lapse is being run on the right with lower autofocus illumination conditions, and we can see that these cells are much happier. They're able to continue proliferating and growing, much like what we saw in that first example on the previous slide. So some considerations in general to keep in mind to prevent phototoxicity is to use the longest wavelengths of dyes that you have available the longer the wavelength, the lower the energy of the light that's going into exciting them, and therefore the lower the phototoxicity will be. And if your instrument is equipped with a laser, near infrared laser, autofocusing laser, then I would highly recommend that you utilize that in place of fluorescence-based autofocus. And then from a setting standpoint, use a relatively low illumination power with higher exposures and gain compared to what you would typically do with fixed cell imaging. This may result in a slightly grainier image, but it will allow your cells to survive these longer time courses. And then generally speaking, your sampling frequencies shouldn't be too aggressive. The more frequently you image, the more light you're putting on your cells, and then the more stress there'll be from that light. So as a general rule of thumb, we tend to start with sampling frequencies of about 20 to 30 minutes, especially if using fluorophores that are excited in the violet and or green channels. Last but not least, I would also like to touch base on different types of microscopes. So what I showed in the last couple of slides was an example of one of our wide field EVOS systems, but many users of course have the need for confocal microscopes as well. And one of the main advantages of a confocal microscope is of course their ability to take optical sections in 3D specimens, but they also provide not only high resolution images due to generally high quality optics, but also very high signal to background by minimizing much of that background by simply eliminating that light from ever reaching the detector. So we talked about a couple different examples of suppressing background using wide field microscopes, such as using the background suppressor and taking out the phenol red out of the media. The confocal microscope itself will actually greatly aid in the suppression of background as well by simply imaging only a very thin volume within the Z dimension, which will essentially reject much of that autofluorescent or background fluorescent light from the media. So many times an experiment that requires a wash and with a wide field system can be performed in a no wash type of workflow using a confocal system. So this is going to be an example of that of using that tubulin tracker deep red once again, but in this case we're actually taking images without performing a wash step using a confocal. So you can see right off the bat that the image quality is actually quite superior in terms of signal to background, where even though we're leaving the label on the cells the entire time, we don't have that drastic background fluorescence outside the cells from unloaded dye, and we're not using any suppressor or anything of the sort here either. Now, one of the other advantages to using these types of scopes is their general sensitivity tends to be much higher. So this also means that you can sample at much higher intervals and frequencies. So now instead of taking 20 images every 20 minutes, we're actually able to take an image every 5 minutes. And these are primary hippocampal neurons, which are relatively sensitive cell type compared to the HeLAs that we were imaging previously. And we can see that we're able to image these every 5 minutes all the way out to about 72 hours without really any induction of cell health or cell toxicity or detachment from the substrate. And this is, again, also duly in part due to the fact that the tubulin tracker deep red is excited by a deep red 638 nanometer laser. So this longer wavelength combined with the higher sensitivity of the detector enables us to not only sample over longer periods of time while keeping the label on board the entire time for free exchange inside and outside the cells, but it also enables us to have much higher sampling frequency without causing any cytotoxicity. Some considerations to think about before moving from a wide field to a confocal is that it may not always be necessary. Oftentimes the confocal instruments in shared facilities will have a cost associated with them, and that cost may not be necessary for the experiment that you're trying to perform. And then even though we have higher sensitivity detectors on many confocal systems, we also tend to have much higher intensity laser illumination. And so this can lead to a greater risk of phototoxicity due to higher intensity laser illumination. So this is where it's generally good practice to use the highest PMT gain or camera gain, depending on the type of detector that you have, using the highest gain setting that doesn't degrade image quality while losing the lowest amount of laser power possible in order to minimize any cytotoxic effects. So in summary, these are the 5 steps that we have talked about today and are kind of the general 5 steps that one can take when it comes to performing live cell imaging. The first step, as we talked about, of course, is to determine if live cell imaging is necessary for your experimental studies. It can be quite a bit more challenging and time-consuming to perform a live cell experiment versus a fixed type approach, and so it is important to really spend some time thinking about if it's necessary, and then also planning out how to execute that experiment. The second step is to culture cells and start with a nice healthy culture that is not overconfluent and that is growing in optimized growth conditions, both in terms of environmental conditions and culturing media. And then really this is where kind of the rubber usually hits the road is at step 3 when it comes to labeling and optimizing and imaging. This is where it's really important to start paying attention to things like concentration and spectral overlap, and really kind of spending some time, again, planning your labeling strategy and choosing the reagents that not only minimize spectral spillover effects but also cytotoxic effects. And so that's where it's important to really spend some time in step number 4 to optimize these conditions and methods in your particular experimental condition, because these things will vary from one cell type to the next. And so these first 4 steps can be pretty time consuming all on their own. And then the last step is, generally one that can be overlooked from time to time, which is the imaging, but it can actually be also relatively challenging to perform the imaging itself, because there are multiple parameters to think of when it comes to using fluorescence in live cells. And one of them, as we've talked about pretty extensively in the previous slides, is not only the effect of cytotoxicity from your label, but phototoxicity from the act of imaging the cells too frequently and with too much illumination. So that concludes this particular 5 steps webinar. For more information about our webinars and similar types of content, please feel free to visit our website for various 5 steps tutorials and more information. And with that, I would like to thank all of you once again for not only your attendance today, but also for any questions that may arise. And so with that, I'd be happy to take questions. Thank you.